Thanks for
okay.
that.
Thank you very much, Carlos. So now we need to move on. And our next speaker is Paul Langton. Paul comes from the University of Bristol, and he's working in the group of Eugenia Pedini. And I think that the main interests are mechanisms behind cell competition in cancer. So Paul, thank you very much for joining us, and all yours. Let me unmute you. Okay.
Thank you. I'm unmuted now. So let me just share my screen. Okay, does that look all right?
Yeah.
Okay, great. So my name is Paul, and I work in Eugenia Pedini's lab at the University of Bristol. And my talk today will be about two proteins called Xrp1 and Irbp18 and their role in cell competition. In Eugenia's lab, we work on cell competition, which is the fascinating process whereby less fit cells are eliminated by their fitter neighbours. The larval wing disc is an established model for studying cell competition, and all the images I show will focus on the pouch region of the disc, which is a simple columnar epithelium. In this example on the right, the GFP-labelled cells carry a mutation that makes them less fit than the non-GFP cells. Competitive interactions between these two cell types causes the loser cells at the border of these labelled patches to undergo cell death, and dying cells in this picture are labelled in magenta. Drosophila is a really good model for studying cell competition because the genetic tools available allow us to easily generate these labelled patches of competing cells and to knock down or overexpress genes of interest within them. So these less fit cells, or loser cells, may arise naturally in the tissue due to mutation or accumulation of damage due to aging. The surrounding fitter cells, or winner cells, recognize these loser cells and initiate signaling events that lead to their removal from the tissue by cell death, and this is followed by compensatory proliferation of the winner cells. It's important to emphasize that cell competition depends on these local interactions between cells of differing fitness, because in homotypic tissues, either of these cell types can survive and grow. Cell competition is thought to be protective against both cancer and aging, as removal of damaged cells maintains tissue health. Several different genetic manipulations can trigger cell competition, but our work focuses on minute cell competition. So ribosomes, of course, are the protein production factories of cells, and they're composed of rRNA molecules and around 80 ribosomal proteins. Minutes are a class of Drosophila mutations in the genes encoding ribosome proteins. And flies which are heterozygous for minutes, or RP+, are viable, but they have shortened macrochetes and are developmentally delayed. And in mosaic tissues, cell death is elevated in RP+ cells neighbouring wild-type cells. In the image shown, the GFP-positive cells are heterozygous for RPS3, and the non-GFP cells are wild-type. Dying cells are labelled by the DCP1 antibody staining in red, and hopefully you can see that cell death is enriched in the minute cells at the interface with the wild-type cells. So I thought it would be good to describe how we do a typical cell competition experiment. So we normally collect the embryos for 24 hours, and then on day 3 we heat shock the larvae to induce many GFP-positive RP+ cells within the disc. Over the next 3 days, the clones arising from these cells will be outcompeted by the wild-type cells and shrink in size, such as by the time of dissection, they occupy a relatively small proportion of the disc. Using this system, we are also able to overexpress or knock down genes of interest in the RP+ clones and assess the effect on cell competition. For example, we can knock down genes by RNAi in the loser clone specifically and see whether the removal of that gene rescues their elimination or enhances it. And examples of what this might look like are shown on the right-hand side of the slide. Therefore, the size of the remaining loser clones is one good readout for how much cell competition has occurred. The other main readout for competition we use is the amount of cell death occurring at the border of the loser clones where they neighbour the winner cells. Shown here is an output image from our custom-built ImageJ macro. We define the pouch area for each disc and the macro segments the clones as shown in the top right panel, and this gives us our clone size measurements for each disk. The macro also divides the clones into center and border territories as shown in the middle bottom panel, which is combined with the segmentation of the DCP1 positive dying cells in the bottom right panel. In this bottom right panel, the dying cells at the border of the clones are highlighted in red, and the dying cells at the center of the clones are highlighted in blue. Therefore, the macro can determine for each disk the percentage of the border or the center that is DCP1 positive. And in a competing situation like an RPS3 loser clone, we would expect an increase in border death relative to center death. And knockdown of a gene that rescues competition would be expected to reduce the number of dying cells at clone borders. We can also study the biology of minute cells in non-competing disks by generating either anterior or posterior compartments that are minute, or RP+. Cell competition doesn't occur across the compartment boundary, so with this setup we can investigate the pre-existing differences between minute cells and wild-type cells in the absence of competition. Our group found that minute cells show activation of several stress response pathways, including the oxidative stress response, which is mediated by NRF2, and shown here using GSTD1-GFP, which is an NRF2 reporter. Minute cells also activate the JNK signaling pathway, which is a pro-apoptotic pathway activated in response to various cellular stresses, and shown here by staining for phosphorylated JNK. So what might be causing the activation of these stress pathways in minute cells? Well, our group and others showed that RP+ cells experience significant proteotoxic stress, which is responsible for activating these stress response pathways. I don't have time to go through the published data in this talk, but we showed that RP+ cells display defects in the protein degradation systems, and the proteasomal and autophagy systems become overloaded, and flux through these degradation pathways is reduced. And this ultimately leads to the formation of polyubiquitinated cytoplasmic aggregates. And remarkably, we found that alleviating proteotoxic stress by inhibiting TOR signaling was able to rescue minute competition, showing that proteotoxic stress is a driver of the loser status in minute cells. It was also shown in these publications that RP+ cells show increased phosphorylation of EIF2 alpha, which is a known response to proteotoxic stress. Increased phospho-EIF2 alpha acts to inhibit global translation and to induce the expression of chaperones, both of which help cells experiencing proteotoxic stress to recover. So I have two more important proteins to introduce before I can move on to my recent results. A few years ago, genetic screens carried out in two different labs identified the bZip transcription factor Xrp1 as a strong suppressor of cell competition. So removal of even one copy of Xrp1 can rescue both the growth and competitive death of minute clones, as shown here where the minute clones are magenta and the dying cells are labelled in green by the DCP1 antibody. It was also shown that Xrp1 is transcriptionally upregulated in minute cells using a reporter of Xrp1 transcription called Xrp1-LACZ. So RP heterozygous cells upregulate Xrp1, which somehow reduces their fitness. Nick Baker's lab subsequently showed that IRBP18, which is known to form heterodimers with Xrp1, is also required for the elimination of RP+ clones. The images at the top right show disks containing GFP-labeled minute clones, and when these clones are generated in IRBP18 mutant background, shown on the right, they're much larger, suggesting that competition has been rescued. IRBP18 is well conserved in mammals, however, there's no obvious homologue for Xrp1. But they suggest at the end of this paper that Xrp1 might be the functional homologue of mammalian CHOP, which has the most similar bZip domain to Xrp1. Overexpression of human CHOP in the Drosophila eye gives a small, rough eye, and this can be rescued by removal of IRBP18 or Xrp1. Therefore, Xrp1 may be the fly equivalent to CHOP, and we'll come back to CHOP later in the talk. So I've described so far how both proteotoxic stress and the IRBP18 Xrp1 complex are required for minute cell competition. But we wanted to know whether Xrp1 and proteotoxic stress act independently, as shown here would suggest, or whether they act in a common pathway. To address this question, we first tested Xrp1 and IRBP18 RNAi lines. Xrp1 expression has been shown to require its own activity and the activity of IRBP18, so we tested whether knockdown of these genes by RNAi in wild-type discs would reduce levels of an Xrp1 transcriptional reporter. Knockdown of either gene in the posterior compartment, which is labelled here with GFP, strongly reduced levels of Xrp1 LAC-Z, shown in magenta, which indicated that these RNAi lines were functional. As a further confirmation, we tested whether Xrp1 or IRBP18 RNAi would rescue minute competition. So these are wing discs with GFP labelled RP over+ clones. Knockdown of either Xrp1 or IRBP18 significantly rescued clone size, which is consistent with the rescue of competition. So now we had functional RNAi lines targeting Xrp1 and IRBP18. We were able to probe the relationship between these genes and proteotoxic stress in minute cells. So first, we tested whether knockdown of these genes would rescue proteotoxic stress in minute cells. As readouts of proteotoxic stress, we stained for phospho-EIF2-alpha, which is induced in response to proteotoxic stress, and assessed levels of the NRF2 reporter GSTD1-GFP, which we've previously shown is induced downstream of proteotoxic stress. So shown on this slide are fully minute discs where we have knocked down either Xrp1 or IRBP18 in the posterior compartment. And knockdown of either gene rescued the accumulation of phospho-EIF2-alpha and GSTD1-GFP, suggesting that these genes are required for and function upstream of proteotoxic stress in minute cells. P62 is an autophagy receptor in cargo which accumulates in RP over+ cells, probably due to defective autophagy, and it's therefore another readout of proteotoxic stress. When we knock down either Xrp1 or IRBP18 in the posterior compartment of RP over+ discs, we found that the accumulation of P62 labeled aggregates was significantly reduced, again suggesting that Xrp1 and IRBP18 function upstream of proteotoxic stress in minutes. We next wondered whether Xrp1 promotes proteotoxic stress in another non-minute loser background. So mahjong is functionally distinct to ribosomal proteins, but like minute cells, mahjong cells are also outcompeted by wild-type cells. So mahjong is another gene which is involved in competition but has nothing to do with the ribosome, or not reported to have anything to do with the ribosome. And interestingly, we found that mahjong clones also accumulate phospho-EIF2-alpha, suggesting that proteotoxic stress may be responsible for their elimination too. When we co-expressed Xrp1 in the mahjong RNAi, Xrp1 RNAi in the mahjong RNAi clones, the upregulation of phospho-EIF2-alpha was rescued. And this is quantified here, and the size of the clones was also rescued. Therefore, Xrp1 induces proteotoxic stress in two distinct loser backgrounds, minutes and mahjong. We then wanted to determine whether Xrp1 overexpression was sufficient to induce proteotoxic stress in wild-type cells. We found that overexpression of Xrp1 in the posterior compartment resulted in very early larval lethality. Therefore, we took advantage of the temperature-sensitive Gal4 inhibitor GAL-ATTS to prevent Xrp1 expression throughout most of development by placing the cross at 18 degrees. Shifting the larvae to 29 degrees 1 day before dissection releases the inhibition of Gal4 and induces a short burst of Xrp1 expression. With these conditions, we found that Xrp1 expressing compartments upregulated P62, phospho-EIF2-alpha, and GSTD1-GFP, suggesting that Xrp1 is sufficient to induce proteotoxic stress in wild-type cells. So altogether, the data I've shown so far suggests that Xrp1 is required for and acts upstream of proteotoxic stress in minute cells. Conversely, IRBP18 overexpression in the posterior compartment did not induce markers of proteotoxic stress, suggesting that unlike Xrp1, IRBP18 is not sufficient for proteotoxic stress. This is consistent with data from the Baker lab, showing that GFP-positive Xrp1 clones are eliminated from wing discs, whereas IRBP18 overexpressing clones are not. Although IRBP18 was not sufficient to induce proteotoxic stress, we found it was at least partially required for Xrp1 to induce proteotoxic stress. So the accumulation of P62 labeled aggregates induced by Xrp1 overexpression was significantly reduced when IRBP18 was also knocked down. The graph on the right shows the ratio of phospho-EIF12 for signal intensity between the compartments. Xrp1 overexpression in the posterior compartment induced a roughly 1.7-fold increase in phospho-EIF12 compared to the control compartment, but only a 1.3-fold increase when IRBP18 RNEI was expressed. This also agrees with data from others, which shows that small Xrp1 overexpressing clones can be recovered in an IRBP18 mutant disk. So everything I've shown so far suggests that Xrp1 is upstream of proteotoxic stress in minute cells. However, we had a clue that Xrp1 might also act downstream of proteotoxic stress. We know that phospho-EIF12 levels are high in minute cells, suggesting they experience ER stress and activate the unfolded protein response, or the UPR. The PERK branch of the UPR shown here responds to ER stress by increasing phospho-EIF12, which inhibits global translation to reduce the number of newly synthesized peptides entering the ER. However, translation of certain transcripts, including the ATF4 transcript, are actually increased during ER stress. And ATF4 is a transcription factor that regulates expression of genes that help cells cope with ER stress, including chaperones. ATF4 also regulates the expression of pro-apoptotic genes, including CHOP, that initiate cell death upon prolonged ER stress. At the start of the talk, I mentioned that Xrp1 might be the functional homologue of CHOP. Therefore, we tested if Xrp1 expression would be induced by the UPR. To test this hypothesis, we increased phospho-EIF12 levels in otherwise wild-type cells by knocking down GAD34, which acts along with PP1 to dephosphorylate EIF2-alpha. Shown on this slide is confirmation that the GAD34 RNEI line we used does indeed increase phospho-EIF2-alpha levels. Next, we knocked down GAD34 in the posterior compartment, labelled here with GFP, and found that levels of Xrp1 LAC-Z were considerably higher in GAD34 RNEI cells. Furthermore, ATF4 overexpressing compartments also upregulated Xrp1 LAC-Z. This shows that Xrp1 expression is induced downstream of proteotoxic stress by the UPR and is consistent with the notion that Xrp1 is equivalent to mammalian CHOP. Having shown that GAD34 RNEI induces Xrp1 expression, we next asked whether high levels of phospho-EIF2-alpha were sufficient for competition. We found that GAD34 RNEI expressing clones were rapidly eliminated from wing disks, and only a few basally extruded fragments of clones remained. To assess whether these clones are eliminated by autonomous death or by cell competition, we used the temperature-sensitive GAL80 again to make large GAD34 RNEI expressing clones. So after clone induction, which is done with a heat shock, the cross was placed at 18 degrees for 3 days to allow the clones to expand without expression of GAD34 RNEI. The cross was then transferred to 29 for 1 day to induce GAD34 RNEI expression, which we confirmed was long enough to induce high levels of phospho-EIF2-alpha. Under these conditions, the wild-type clones showed very little cell death. GAD34 RNEI clones, however, contained many dying cells. And importantly, cell death was higher at clone borders than at clone centers, showing that high levels of phospho-EIF2-alpha are sufficient for competition. Having established that GAD34 knockdown cells induce Xrp1 and are outcompeted by wild-type cells, we asked whether this competition was Xrp1 dependent. As I showed before, without GAL80TS present, GAD34 RNEI clones are completely eliminated from the wing pouch. When the clones also express Xrp1 RNEI, they're recovered in every disc, suggesting that Xrp1 also acts downstream of proteotoxic stress. Altogether, our data suggests that proteotoxic stress in minute cells induces Xrp1 expression downstream of phospho-EIF2-alpha, and then Xrp1 acts along with IRBP18 to induce further proteotoxic stress in a feed-forward loop. Much like minute cells, GAD34 knockdown cells upregulate GSTD1 GFP, and we found this was fully dependent on Xrp1. Remarkably, Xrp1 knockdown also partially rescued the increase in phospho-EIF2-alpha in GAD34 knockdown cells. This supports the feed-forward loop model depicted because it suggests that some of the phospho-EIF2-alpha generated upon knockdown of its phosphatase is contributed by Xrp1-induced proteotoxic stress. So we previously showed in a publication that NRF2, which is the master regulator of the oxidative stress response, is activated by proteotoxic stress, and that expression of NRF2 is sufficient to induce competition. Therefore, we asked whether Xrp1 is also responsible for the elimination of NRF2 overexpressing clones. When we generated NRF2 overexpression clones, only a few tiny clones remained by the time of dissection. A knockdown of either Xrp1 or Irbp18 rescued the growth of these clones significantly, suggesting that the elimination of these clones also requires Irbp18 and Xrp1. It remains to be established whether additional target genes, additional NRF2 target genes also contribute to the loser status in NRF2 overexpressing cells. So we next wondered what could be the initial source of proteotoxic stress in Rp over plus cells that triggers this feed-forward loop I just described. Our lab and JP's Vincent's lab showed that minutes have a stoichiometric imbalance in their ribosome subunits, and we suggested in the previous publications that this was the source of proteotoxic stress. The red dots on this graph show the abundance of ribosome subunits in RPS3 wing discs relative to wild-type wing discs, as determined by mass spectrometry. As you can see, RPS3 heterozygous discs have a reduced complement of small subunit proteins and an excess of large subunit proteins. Given the abundance of ribosomes in eukaryotic cells, it's easy to imagine how unassembled ribosomes can overload the protein degradation machinery and cause proteotoxic stress. Interestingly, we found that removal of one copy of Xrp1, which is sufficient to rescue minute competition, largely prevented the increase in large subunits, but the levels of small subunits were still reduced. Therefore, it seems that in Rp heterozygous cells, there is an initial Xrp1 independent imbalance in the ribosome subunits caused by a reduction of small subunits, which is likely to induce proteotoxic stress and Xrp1 expression. Xrp1 then causes further proteotoxic stress, and this somehow leads to the accumulation of large subunits. So what might be the relevance of this work to human disease? Well, ribosomopathies are a diverse class of disorders caused by loss of one copy of an Rp gene, and symptoms include increased cancer risk, reduced numbers of blood cells, and growth defects. And a ribosomopathy patient-derived mutation in RPS23 was engineered in flies and causes increased autonomous cell death and proteotoxic stress. And this increase in autonomous death was found to be dependent on Xrp1. So here Xrp1 is knocked down in the posterior compartment, and you can see there are fewer DCP1 cells. So our work suggests that removal of Xrp1 would also rescue proteotoxic stress in this mutant. And that by, and that by extension, CHOP or regulators of CHOP could be promising targets for the development of treatments for ribosomopathies. Furthermore, removal of Xrp1 has been shown to rescue pupil lethality, motor defects, and gene expression changes in a Drosophila model of ALS, which was very nicely introduced 2 talks ago. And ALS is characterized by the accumulation of ubiquitinated cytoplasmic aggregates in motor neurons. So this raises the possibility that CHOP may be involved in ALS, and that feed-forward signaling between CHOP and proteotoxic stress may contribute to the neuronal toxicity in proteinopathies such as ALS. So I think I should be well within time. So I just need to thank Eugenia. She's a fantastic supervisor. I want to thank all the members of our group for feedback on this story in lab meetings, etc. I'd like to say thank you to everybody for listening, and thank you to the organisers for giving me the opportunity to present my work. And I'm happy to take questions either now or later on the Slack channel. Thank you.
Okay, thank you very much, Paul. That was very inspiring and very nice talk. So there are actually some questions here. So first of all, they were asking how conserved are these mechanisms from Hamed? How conserved are these mechanisms in other tissues of the flies? And what's the implication of the proteotoxic stress for cancer? You have answered a little bit the second one, but how conserved are these mechanisms in other tissues rather than just the wing disc?
Yeah, so I think the iDisk is another established model for cell competition, and I believe in the iDisk, many of the effects we see in the wing disk would be the same. It's not the cell competition between neurons, for example, is not particularly well established, but it's not impossible that it could also occur in neurons. And certainly some of the cell biological changes we see, such as accumulation of protein aggregates, could also be happening in neurons and could contribute to increasing Xrp1 expression there. But we haven't looked, we just used the wing disk as our system.
Okay, thank you. So another question from Jose Teles is there any idea on how Xrp1 could be influencing ribosome subunit expression?
Yeah, that's a good question. No, basically, we don't know the answer to that yet. It's not clear whether there's already a kind of imbalance in the ribosome subunits, and then Xrp1 induces further proteotoxic stress, which overloads the autophagy and proteasomal systems even more. And then, these unassembled complexes can be dealt with even less well by the cell, which leads to the accumulation of large subunits, or whether even Xrp1 has some direct regulation of ribosomal subunits is not known yet.
Okay, thank you. And another question is that you showed that Xrp1 was sufficient, but not Irbp18. So yeah. Do you know if Xrp1 can induce Irbp18 expression?
Yeah, it can. They actually regulate each other's expression, and they're both required for expression of each other in minute disks. So the thinking behind the fact that overexpression of Irbp18 doesn't induce proteotoxic stress or elimination of clones, whereas Xrp1 does, is that the levels of Irbp18 aren't limiting for Xrp1, but the levels of Xrp1 are limiting for Irbp18. So it doesn't matter how much more Irbp18 you put, there's not enough Xrp1 there to bind to it and have these effects. That's one interpretation of those results. Okay,
thank you. So now you showed with this loop that actually the loser really wants to die, because it doesn't only activate, but it makes a loop to make sure that it dies totally. Okay, well, those are a great talk. Thank you very much, Paul. And if there are no more questions here on Zoom, as I say, you can always post most questions in Slack. We will move to the next speaker. Thank you, Paul.